right, for the next exam, there will be several chapters. This chapter is chapter 11, and here are the main concepts. So I'm going to give you some hints as to what I'm going to emphasize on the exam here. So let's start with just this first kind of rundown of what this chapter is about, and I'll tell you which of these, a potential way to study for it. There will be questions on how they identified the genetic material. Now, the genetic material, of course, is DNA, and in general, you need to recognize how they did that. So if you can tell me a story about how they identified that, and we'll be talking about that as we go through these slides. This is one of the things, but it still is just one question. There'll probably be one short answer on this. So, but you need to read it to be able to tell me the story at all. So you need to read it. How much detail you want? Well, let's get there, and we'll talk about it when we get there. Lots of questions over these two, but they're probably not going to be essay questions. So they're not going to be worth a lot of points individually, but collectively they will be. So you have to know all about the DNA structure, double helix, base pairing, that sort of thing. What does it mean to be complementary? All of those terms, and we've talked about those already in videos I've posted. We'll just go over it a little bit more detail here. So lots of like multiple choice, true, false, that sort of thing on nucleic acid structure. Now, the discovery of the double helix, there can be some questions on there, but again, they're going to be multiple choice and true, false. I would read it, but as far as having to tell me the story, you probably wouldn't have to tell me the story. You might have to tell me a little bit up here about the identification, though. So these two, actually, they're worth a lot of points, but it's mostly going to be true, false, multiple choice. So that's kind of the level that you need to study this for. So you'll need to be able to identify those. It's not like these aren't worth a lot of points. They are, but you know, just focus on learning the basics and reading through it. And if you've done this before, you probably know most of the things about DNA structure, and I'm going to try and point out some of the things that I figure you may not know about DNA structure, especially when we get down to this last part. Those are probably going to be the things I'm going to focus on that are worth more points. So, like I said, it isn't that these two are not important. They are. They're just not as important as the things that come later. Now, overview of DNA replication. This is an important one. If you want to get an A, this one tends to be the thing that kind of distinguishes between getting an A or a B on this exam. This is going to be a large question. It's very complicated, and I've got to admit it is not easy to understand at all. I'll do my best. My students usually tell me I'm really good at explaining this when I teach it in person, but it is a whole different animal when you're trying to teach this online. I'm going to try, and you need animations, all right? And I'm going to try and find some of the better YouTube videos out there that show this, and I'm going to try to help you through this, but there will be a large, high point essay on replication, and that usually will be the thing that will put you over the top for getting an A, if you know it. That doesn't mean it should be the most important thing you study, because if you study this and get it right, but you miss something that is even more important, then you can end up with, you get this right and still end up with a B or even a lower B, because the points here. So this is worth a moderate amount of points, but there are things that are worth a lot more points. So keep that in mind, but this usually is one of the things, even good students can get this confused. All right. Now, the molecular mechanism of DNA replication, well, that goes along with it. I should have grabbed these two as essentially the same thing, and we'll go over those. Now, this one, the molecular structure of eukaryotic chromosomes. I emphasize this a lot. There will be lots of questions about this, and I will give you some of my own videos on this. This should tell you that I consider this very important. And it is because I want you to know the things that are coming up technologically and to understand the latest ideas, the things we're going to kind of cover towards the end that I would like you to understand. You're going to need to understand this piece. So I will be asking lots of things about this. Is this the most important here? Not necessarily on this test, but if you do not understand this, it's going to make it much more difficult on later exams. So please do not ignore the molecular structure of the eukaryotic chromosomes. Okay? Now, so we got that out of the way. Let's go through this. A lot of this you've already reviewed in other videos I've posted. So DNA, I think everybody knows what DNA is. This isn't telling us anything that we don't know here. Necessary criteria for the genetic material. If I ask you this, and I probably will ask you in some way, you need to know the history a little bit. You need to know not necessarily when they do it. I don't care if you understand exactly when they discovered these things. That's not what I want. What I want you to do is understand the order. What did they discover first? And why was it important? So the biochemical basis of heredity, and when they came up with this idea that, hey, the chromosomes must be the thing that's carrying it. What is it about chromosomes that makes you believe that? And of course, if you go back and read some of the things we've already read, it's because you get some of it from one parent and a little bit from another parent, and they combine to make the new one, the new child or the new organism. And of course, that one has traits that come from both. So if you remember all the Mendel stuff, it kind of leads over to that, hey, these chromosomes must be the thing that's carrying the basis of heredity. So keep that in mind. Now, protein was an alternative to DNA, and to tell you the truth, that wasn't a bad idea, and today we're kind of looking at this, and there may be more to that than we thought. Kind of like the guy we talked about last time. What's his name? With the giraffes. I can't believe I can't remember his name because I really like the guy. Oh well. But the same idea. There was an old idea that maybe proteins and not DNA were the carrier of heredity. And it was laughed off later on. It says, nope, that's not it. It's all DNA. Everything is DNA. And of course, now, these days, we're learning, maybe there is something to that. And of course, it's related to this epigenetic stuff that I mentioned before, related to I still cannot remember that guy's name. It's going to bother me all day now. All right, well, we'll go on. I'll remember it probably as we get closer. Lamarck, sorry. So let's move on. Now, bacteria are not the same as us, but they do have traits that are similar, and you need to know maybe this guy, Frederick Griffin, and he did some experiments, and there was a smooth-looking one and a rough-looking one. Didn't know exactly what all this was, what the capsule shield of the bacteria was or anything. He didn't know exactly what these things did, but he knew that roughly that they were traits, and that they acted maybe not like traits that we learned in Mendelian genetics, but he knew they were inherited. And he did this type of experiment, all right, and you need to know this experiment. I am not going to go through it. You can go find videos on it on YouTube if you want. But if you read it, it's very straightforward. I would probably ask you a single short answer on something like this, on Griffin's bacterial experiment. He, some small piece of what he found, why it was important, that kind of thing. Or maybe, what is transformation? Maybe that's a good multiple choice question. So read over this, learn what he did, try to understand why it's important. All right, you're teaching yourself on this one. And so I'm going to skip forward. Now, on Avery, Mcleod, and Mccarty, they're the ones who really convinced most people that DNA was the thing. So you need to understand what they are. Now, I will tell you this, something to think. DNase and RNase are enzymes. Those are proteins, and they're proteins that cut or destroy whatever there is before the ace part. So DNase, all right, kind of is, it destroys DNA. RNase destroys RNA. So and proteases destroy proteins. So the idea here is they used these in this experiment, and that's what I would focus on. How did they use these things that destroy specific things to figure out that DNA was indeed the genetic material? All right. You go ahead and read this, get the idea for it. Now, we've already talked quite a bit about DNA and proteins and lipids and things like that. I will be making a couple of new videos, one of them on water. I promised you a video on water, and then I'm going to also give you a video on the harder part that we talked about called replication, and we're not here yet, but do expect 2 videos coming up that are mine on why water is special, and also in the properties of water, and also on replication. Now, what that means, if I make a video on it, probably means I'm going to ask more than multiple choice or true-false questions on it. So when you watch the videos that I make, realize I'm kind of telling you that, hey, there's going to be something more than just a multiple choice or true-false question on this. All right, you do need to know these terms. All right, now these make good multiple choice and true-false questions. All right, we need to know that DNA is wound up. It's a double helix, but that double helix also winds up into the chromosomes. Now, this is our first chance to talk about them, but they interact with proteins. There are these proteins called histone proteins that they wrap around, and we'll get more into that in a second, but they are very important for that stuff I called epigenetics, and we will get to that. But these proteins called histone proteins clump together, and then the DNA will wrap around these histone proteins and form what's called a nucleosome. So we have, this doesn't show it here, but see, we have DNA wrapped around this cluster of histone proteins. There are actually many of them in the little beige part here. And the DNA wraps around them, and we call that a histone. So I'm setting you up for the things that you need to know. And then these all cluster together and form the chromosome. And we'll get to better pictures than this one after a while. But the whole point here is you're seeing that it's individual nucleotides, ACGTs. They come together to make a single strand. Then the single strand comes together to form the double strand, the double helix. And then that kind of coils up like a telephone cord, an old style telephone cord. And then that then wraps around these histone proteins. We call it a nucleosome when it does. And then those all compact together to form a chromosome. You need to know all of those things I just said there. All right, we've already gone over this. Maybe not in the detail they have it here, but you do not have to learn the chemical structures. Some professors make you learn that. I do not. What I need you to know are 3 things. There is a base, and that's going to be either the A, G, T, or C. So one of those 4 bases, and it's connected to a deoxyribose, which is also connected to a phosphate. So you need to know there's a base, the ribose, and the phosphate. Now, ribose is a sugar. So deoxyribose, deoxy means it doesn't have an oxygen. There's an oxygen missing. But it's a sugar that you might think is missing a sugar, missing an oxygen. Sorry, it's a sugar that's missing an oxygen connected to what we call a phosphate on one end and a base on the other, and the base can be one of 4 things in DNA. Now remember, in RNA, which we've talked a little about, you replace the T with a U, so it's uracil. So there are 5 bases that we consider in nucleic acids, but in deoxyribonucleic acid, which, remember, deoxy, this is DNA, all right, there are 4 bases, and these are the 4 for DNA. So that's kind of stuff I can ask for true, false, multiple choice questions that can confuse you. I might say, give me the 4 bases in DNA, and I might go A, G, C, U. Well, that would be an incorrect choice on a multiple choice. Now, where do we use U? We use U if it's RNA, and RNA isn't deoxyribose, it's just ribose. So it's RNA instead of DNA. All right, so you need to follow that. That's what they're going to go over here in this next one. And so you notice this is just ribose. So this is where the R comes in for RNA. Deoxyribose, that's a D, so it's DNA. So there's a chemical change here. Deoxyribose and just ribose. And then, of course, the 4 bases are a little different. The phosphate is the same. Now, there is a sugar. Ribose is a sugar. Deoxyribose is a sugar. They're just slightly different sugars. All right. The bases, most of them are the same, but you replace the U and the T there. And the phosphate is the same. So that's the same as the one up here. All right. So keep that in mind as you're learning those. Those are where I can kind of, give you some oddities in the multiple choices, making sure that you know the difference between DNA and RNA. You should know how they're numbered. They're numbered by where the attachment starts. So see this long attachment, so it attaches here, 1, and then you go 2, 3, 4 around this way, 1, 2, 3, 4, 5 around this way. We don't count this guy. So if I give you a picture or something, I would ask you, what is that 2 on the base? What is that position 2 on the base? You would say, oh, that's a double-stranded oxygen right there. All right, if I ask you what is at 5 on the deoxyribose, you would go, oh, that's a CH2 that connects to the phosphate. Okay, so those kind of things. So you do kind of need to know what's at. So there's nothing really interesting. There's an OH here. Now compare that to the DNA. OH is there. Let's see, where is this oxygen that's missing? OH. Check that out. Make sure you know what's going on there with this deoxy versus oxy. All right, now here we go back, and of course we have our base pairing. This is a single strand. Notice that the phosphates line up here, the sugars line up here, and that the bases stick out. And that's because they're going to bond with the other strand of DNA. So if this is a T, then over here you're going to have this in reverse. You're going to have the phosphate, you're going to have the sugar, and then of course if it's going to line up over here, this is going to have to be an adenine, an A, because this is a T. So that's going to be what has to be there. But on the other side, you're going to get what? You're going to get the sugar and the phosphate. They're just going to be in reverse. So sugar here, phosphate on the outside. All right, and that's going to form your double helix. We also read them in a particular direction, 5' and 3', and that has to do with where the ends are. 5' is the phosphate side, all right, and this 3' is the OH side on the sugar. So this is the 5' end of the DNA, and this is the 3'. Now something you need to know is over here it's going to be reversed. If you're 5' here, you're going to be 3' over here, and 5' at the end. So know that that's supposed to be a 5 there. So how did I end up with a J? There we go. 5', and that means this side is ending with a phosphate, and this side's ending with the sugar. All right, those are the ones sticking out on the ends, you might think. All right, and that gives it directionality. So you need to know that term, directionality. Features. Know most of these, or at least something about each of these. Know what base pairing is. Know what a hydrogen bond is. We just talked about the sugar phosphate backbone. The backbone is the stuff besides the bases. So here in this red stripe, that's the backbone. In our case, this is the backbone. See, they put these chemicals in the red stripe. That's what's representing the backbone. Know what it means to be right-handed. know what it means to be anti-parallel and double-stranded. And I think everybody knows what double-stranded means. It just means you're bonding. You've got, here's one strand and here's the other strand, and they're stuck together. All right, DNA structures. We've already gone over this in other videos. Understand this. Now, which of these bonds do you think is strongest? Well, if you'll notice here, here's a C to G, and here's an A to T. So those are our two types. It can be A binded to T or T to A. They're the same thing. But notice there are only two, and these are hydrogen bonds right here. They're not very strong, but there are only two of them. But over here, when you go G to C, there are three of them. So which of these do you think is stronger? Well, it's the one with three. So a GC bond is stronger than an AT bond. You need to know that. All right, and when it all comes together, you need to know that there's something called the minor groove and the major groove. Proteins tend to prefer binding to the major groove. Doesn't mean that they all do. It very depends, but the majority will choose to bind to a major groove. This brings in the idea that the DNA is interesting. The structure has a tendency to attract proteins. So here's a little protein I'm drawing here. And the proteins will be shaped, all right, to fit into the major or minor groove. Now, the majority of them fit into the major groove. But they'll fit and they will stick. So they will kind of stick in here, all right, and attach and bind to the DNA. It's very important. This is important for things like transcription and translation and replication. You have proteins sticking to this, doing things to the DNA. But it's also important for gene regulation, for turning them on and off, that we will talk about much later. But you need to know that proteins, people think of the DNA all by itself. No, no, no. There are proteins stuck all over the DNA at all times. Only in the laboratory, when you add chemicals to make the proteins fall off, is the DNA alone. Most of the time, it's like there are things just stuck all over this thing. It's like what would be something that normally if you see it in a museum, you know, it's all cleaned and polished, but in real life, it's got stuff all over it. Like maybe a ship. A ship has barnacles. Things stick to the hull of the ship. And that's what the ship normally looks like, but you know, they clean it up and put it into a museum. It's all clean and you know, like it was just made. Well, that's the way DNA is. DNA, when it's being used in your cell, just has proteins stuck to it everywhere. You don't have to worry about this. You do need to know that about Rosalind Franklin and the X-ray diffraction. I'll ask some short questions on that, either true, false, or multiple choice. Read this. You need to read it in general. We already know Chargraph's rule. Chargraph's rule just says A's bind to T's and C's and G's. So what does that mean? That means you essentially have, if you have a certain amount of T's, you're essentially going to have the same amount of A's. And whatever amount of C's you have the same amount of G's. Really not that different. He went and just looked at this and how it was different in different organisms. But his rules were that this ratio is the number of these equals the numbers of these. He's not saying that much, though it is interesting. But he looked at this, and of course, what does it show? A's bind to T's. Well, are they the same? They are. It's just bad measurements. There are regions in the DNA where the double helix, there are regions that don't bind together. So it should be off a little. But in general, you should see similar ratios at 1-to-1. It's just that they're not as good at getting these numbers. They're not as accurate as they could be. There are limitations to the chemistry. Know who Linus Pauling was? Linus Pauling was really amazing. He did lots of things in biology, chemistry. He had multiple Nobel Prizes, I believe. But you should know what he did. And Watson and Crick are the ones who are attributed with discovering that indeed the DNA has this double helix structure. and Rosalind Franklin also was involved. Let's see. So it really should be the three of them, or if you include Maurice Wilkes, there should be four of them. But these are the two guys that are famous for discovering the double helix structure of DNA and why it was so important. Right here is their first model. This is what they said it looked like. Now here we get to the overview of replication. I'm going to make a whole video about this. Do your best to figure out what they're talking about in the book. I will definitely ask you about how they figured out if DNA was semi-conservative, conservative, or dispersive. Now, I won't talk about this in my video. You need to read this and learn this yourself. Am I going to ask? I might ask one question on Matthew, Mendelssohn, and Stahl. And that's a maybe. I'm not even sure I will ask that one. DNA replication. Now here's the hard part that I'm going to make most of my video on, or I'm going to go find better ones on the internet than what I can make. But DNA replication, it's that thing I talked about at the beginning, that it's one of the things that's difficult to understand, and I'm going to ask a large, important question on it, and it seems to be the question that, distinguishes between an A student and a B student. So, if you're trying to make an A on the next exam, make for sure you go and try to understand replication, but don't do it at the expense of other things, because there are other things that are worth more than this one. This one is just something that's worth, 10 points or so, and it's hard. There are other things that are not as hard that are worth, 20 points. So, don't not study something else just to study replication. But once you're done, make sure you're good on most of the other things I emphasize. But there will be a question on DNA replication, and it'll probably be worth about 10 points. All right, now, what is the hard part? Now, this part is not hard. You can read this, and I think you can get it. The hard part is going to be down here when we start talking about these proteins that do the work. This is the part where it starts getting hard. And you need to remember that ATP is your power source. Just as a throw-in, that's something that definitely need to know in my class. I emphasize the importance of ATP. And what does ATP do? It powers your proteins. They can change shape. You add some ATP to a protein and it changes shape. And when it changes shape, it's doing work inside your cells. Now that could be chemical work or physical work. It can move something or it can chemically alter something. But you need ATP usually to perform that work. And of course you get ATP from glucose. Your mitochondria or your chloroplasts, at least in eukaryotes, their function is to convert glucose to ATP. Bacteria don't have those structures. They're very simple, and they do it just in the cytoplasm. All right, so here's where it starts getting hard, and you realize you have to unwind the DNA. So I'm going to be either finding really good videos for this, and maybe my own video. I've made them before. Like I said, my students usually tell me I'm really good at explaining this in person. We'll see how well I am online. But the hard part of this is first you have to understand you have to unzip the DNA. And so there are proteins that are going to allow you to do this. So the Topo Isomerase here kind of straightens the DNA to get ready. Remember, it's kind of twisted. It's a twisted ladder, a double helix. So you have to have something that straightens it out. And then you have to have something that basically makes it unzip. And so here you have a couple of proteins that are going to do those things. This is not hard so far, but this is the first step. DNA polymerase. Now there's something called RNA polymerase and DNA polymerase. Do not get these mixed up. Polymerase just means, if you want to know what this means, it means copy machine. It's a photocopier of DNA, if you want to think of it that way. Now, of course, what does it make when you copy? Well, DNA polymerase makes a DNA copy. So think of it this way. DNA is like a color photograph. all right, or color picture. And when you use DNA polymerase, you end up with another color picture. All right, you're essentially making another double-stranded DNA when you use DNA polymerase. However, this is a polymerase. There's a protein here that does this, and that's DNA polymerase. So you have DNA, you add DNA polymerase, it makes more DNA. But there's also RNA polymerase, and in RNA polymerase, all right, so I changed just one letter. I went from DNA to RNA. When you add that one, okay, when you add that protein, all right, that one, the result is RNA. And you can think of RNA as a black and white picture. Or maybe better yet, think of it as double-sided paper. Either way, it's not the same quality as the original. So maybe you think of this DNA as double-sided or double-stranded. All right, so your paper is double-sided, and when you use DNA polymerase, you end up with something that's double-stranded or double-sided, as in a copy. And then over here, it'd be like having a copy machine that only makes single-sided copies. So that's one way to do it, or it makes black and white copies. They're not as good as the one over here. Either way, you have to understand the differences between the DNA polymerase and the RNA polymerase, at least get that part right. But once you are single-stranded, that's when the polymerase can attach. So the polymerase can attach here, and we're going to see that depending on where you're going, you're either going to attach right near where it unzips, or you're going to attach on the other end, and that's going to be what makes this difficult. And I may have put those on the wrong sides there. I hope not. I don't want to confuse you. Let's see. So let's keep going. All right, DNA polymerase only works in the 5' to 3' direction. So that's the direction that it moves. Now we talked about that ends. So let's see if I did this right. So yes. So if this is 3', this is the 5'. So it moves from, as it said here, 5 to 3. So what is it going to do here? This one is going to attach here because that's the 5' end. If this is the 3, then this is the other end, and it's going to attach here, and it's going to move that way. All right. This one is the 5' end is over here, and it's going to move that way. And there are some problems with that. That's going to affect how the rate at which this thing can operate. And one of these is going to get one of these strands is going to get duplicated at a different rate than the other one. And that's going to lead us to the one that goes fast is called the leading strand. And I think that's going to be the bottom one. And the one up here is going to be the lagging strand. And so that means it's a lot slower. And you have to understand why one is slower than the other. And that's going to be the hard question that I'm going to try to help you answer in your little essay. So we're going this, and here you go, leading and lagging strands. All right, and there's this thing called an Okazaki fragment. I don't want to get into it here because it needs to have its own video. But that's going to be the hard part, understanding why you need the Okazaki fragment in the lagging strand. If you can figure out why you need this for this, then you've got it. You're going to answer the question correctly. So focus when you're studying on this. Why do I need an Okazaki fragment on the lagging strand? You don't need this thing on the leading strand, but you do need it on the lagging. So look out for that video, and I will do my best to explain it to you. You can try it here. Try to understand it as well as you can before you see mine. Read it. Try to figure out what's going on. All right. Realize there are different types of polymerases. We're not going to worry about it. We're just going to call it for now DNA polymerase. But really the one we want is DNA polymerase 2. That's usually the one we're... well, no. It can be any of them for what we're doing right here. Let's see. Know the proteins and what their function is. Good for multiple choice, and for describing what's going on, you're going to need to know those terms, these proteins, to tell me, well, this one is doing that, and that's causing this, and that's why we have an Okazaki fragment or something like that. Be able to phrase things that way. read this. This could be multiple choice. All of these little things could be multiple choice or true false. Know that there are a bunch of different DNA polymerases. Know that when you go to bacteria, they rename them. For you do not have to know details here. If you ever take a more advanced cell biology course, they'll make you memorize these things. Just for us, know that they are slightly different, and they have different names. And so an alpha instead of, say, 1, or beta or gamma instead of 1, 2, 3, 4, and 5. So just know that they're there. Know that there is something called DNA repair. In other words, you get a damage on the DNA, the polymerases can come along and fix it and repair the DNA. So just know in general that is one of the functions of the polymerases. Now at the end of the chromosomes, so you have a chromosome, and the ends of them, you have something called telomeres. And we've actually kind of figured out what they do. Essentially, every time you copy a chromosome, so every time you do DNA replication, make a new cell, and you make a new chromosome, you copy it. So you make a copy of it. The ends get cut off. It's kind of not complete. It's like you made a copy of a book, and every time you make a copy of it, you leave the last and the first page off. And of course, eventually you don't have the introduction or the ending anymore. And of course, if you get up high enough where you've cut enough off that you start cutting out genes, well, the cell usually dies. And it's pretty simple, but they're important, especially if the cells die. So every time, it's kind of a built-in aging mechanism in your body that if we could undo that, maybe we would live a little longer. And in fact, they've done that. They've improved the system and actually found some chemicals that can actually keep the telomeres from being chopped off at the end. And sure enough, the mice they do those experiments on live longer, and they're healthier. So that's something we might be working on in the future to make us live a little longer. Actually, they've done several things. If you want to know some trivia, they have mice that if they did the techniques on them that they did their equivalent age, and if you were to transfer that to humans and calculate it, they could make humans that live over 200 years old. In other words, they can actually engineer, add a few genes, re-regulate a few genes, feed them the right things, and they can get these mice living to what would be equivalent to about 200 or more, a little over 200 years in human years, if it worked exactly as it did in mice. No one knows, but it would obviously make some difference, and one of those things is improving this process where the telomeres get cut off. If you're interested in that sort of thing, you can look up telomeres and read about them on Wikipedia and things like that. All right. This explains why they get cut off here. I'm probably not going to ask you that, but you might have a true or false question here somewhere on telomeres. So I would review it. You certainly learn this. If you ever take molecular biology, this will be in that course. And of course, telomeres are related to cancer as well. You don't want them deleting genes. That will confuse the cell. And we've talked about in a previous video how cancer is related to being a confused cell. Now, here are the things that I will definitely be asking you, and you will need to know further in this course, maybe not on this exam, but in future ones. So please make sure you understand what chromatin is. I mentioned nucleosomes already. They wrap around these things called histone proteins. So you're going to need to know that. Know that each histone consists of 1, 2, 3, 4, 8. Okay, it's an octamer. 8 histone proteins kind of stuck together that stick to the DNA also. They kind of surround the DNA. There is one called a linker. Not important that you know that. And know that it takes about 150, a little less than 150 base pairs to wrap around, so 150 letters roughly, a little less, to wrap around one histone. All right, and that's what we have here. We show this picture. This picture I will come back to probably many times to try and explain the function of this. And I will give you a hint right now. What's so important about this? Well, it's how loose or how tight you wrap around this thing. Or if you're in this region where you're not hooked around this. Because if the DNA is wrapped around this thing really tight, essentially nothing else can attach to it. So essentially, you usually downregulate or even turn off completely certain genes. So if there were a gene right here and it got wrapped around one of these, well, that gene is no longer used in that cell, all right, or it might not be. And so, of course, we talked about different cells. How do you distinguish different types of cells in your body? Well, it's by what genes are turned on. So how do you determine what type of cell you are? Well, it depends on what part of your DNA is wrapped around these histones. That is a very, very, very important concept, all right, to understand. In fact, there's going to be an essay question on this, okay? Understand the function of histones. By wrapping around these things, you can regulate the genes. In other words, you can turn genes on and off by how tightly you wrap around this thing. Or if you're just open, of course, this is a gene. Obviously, if there's a gene in here, it's on, all right? Not always true here, it may be on or off, but if it's wrapped really tightly, it's off. If you wrap it really tightly, it means that gene isn't on, you're not going to make that protein, and you're probably making a protein for a particular cell type. In other words, the cells need to be open, all right, for them to be on. They need to be out here for sure, but depending on where you wrap the DNA around these histones, what part of the DNA gets wrapped around these things, pretty much determines what type of cell you are. And that is the important thing you need to know for an essay question on the next exam, on this exam. So remember, these are very important, all right, for getting those questions right. Understand the function of these histone proteins and understand that they are the key to turning on and off genes in a cell, and therefore determining what type of cell you are. All right, here we're just looking at a electron microscope. Electron micrograph is what they would say right here of the fibers. So this is how they know it structures itself like this. Know that it winds up, and there's a special sequence of letters that is what controls if these are really tightly compacted into this structure, packed up. These are packed up like you're packing up a car. You certainly can't use these if they're packed up like this. They're packed up in a back of a truck, and you can't get to the whatever it is, your phone. Your phone gets packed away into a suitcase, into the back of a truck. You're not getting to it, all right? You cannot use it. So it is off and unusable at any point. And that's kind of what controls that. We kind of know it's this guy. All right. It can also lead to pieces that stick out. So if you use this CTCF, you can have a loop come out loose. And hey, if it comes unwound, you know, it's wrapped around here, but then you kind of like give me an extra loop in here. Hey, now there's a region here where you could have a gene that's turned on. So just realize that's kind of how this works, depending on how this special sequence in the DNA is used. All right, Euchromatin, not as compact. So therefore, fewer proteins stuck to it more or less, or Euchromatin, or at least not, fewer proteins in the terms of histones. So this would be the stuff maybe in between, the stuff that I just drew here, all right, might be considered euchromatin. Heterochromatin is much more. Hetero meaning different. It's got DNA and proteins. Hetero, different. Heterozygous, different. Big A, little a, that kind of thing. So heterochromatin means it's made of DNA and proteins. Euchromatin is usually mostly DNA, just a few proteins. There really isn't much of the DNA that doesn't have at least some kind of protein sticking on it. But this has much less compared to the heterochromatin. And this one with a lot of proteins are usually off. I said usually. It's one of those things where you never say never. There are heavily packed, compact genes that are indeed still function, but they are rare. All right, so here we're kind of looking at what I was talking about here, and here is our chromosome starting out with our double-stranded DNA, double helix there. So just keep in mind, review all of this, watch my other videos, and read it. And we're actually done with this one. We got through with this one quicker than I thought. This is something like, what, 73 slides here? Not quite. I took a few of them out. But these are the things you need to work on in Chapter 11. And I will make some videos of my own soon about water and about replication. I may not make one of my own on replication. I may go look for those with some really good videos that are pre-made on YouTube for you to watch. But I'm thinking I will make my own. It's just, we'll see if mine are any better than the ones I found out there on YouTube.